All right, let's continue on with learning about cellular respiration. In this part, we're going to learn about the citric acid cycle. And when you're done with this, you're going to understand this cartoon that says, I may look like I'm not doing anything, but at the cellular level, I am quite busy. And you may be a little bit exhausted. Fortunately, we're not going to learn all of it. We are going to remember the structure of the mitochondrion. Here's our outer membrane, our inner membrane, the matrix inside, and then the intermembrane space. Remember that inter means between. So the intermembrane space literally just means between the membranes. The cristae are these little folds that kind of make like pancakes inside the mitochondria. And then inside the mitochondrion, this is kind of cool, you've got little circles of DNA, just like a prokaryotic cell. We've got other inclusions and things like that. And then embedded in the membrane here, we're going to have the enzymes for the electron transport chain and ATP synthase. And we'll come back to that. This stuff for the citric acid cycle is going to happen in here with these little inclusions and things inside the matrix. The Krebs cycle occurs in the matrix. I like that one. Okay. All right, so here it is. Just memorize this. Yeah, no, I can't either. It gives me a headache, to be perfectly honest, when I see it. There's so much, here. So let's break this down a little bit. And let me talk about what I do want you to actually know from this, okay? So we've got, here's the pyruvate, the result of glycolysis. We already talked about these prep reactions that happen to create acetyl-CoA. Here is the acetyl coenzyme A. Okay, that then is going to hand off acetyl-CoA to this 4-carbon molecule here, oxaloacetate. You do not need to know the name of it. You just need to know it's a 4-carbon molecule. The acetyl-CoA, remember it's a coenzyme, hands off the acetyl part to this 4-carbon molecule to create citrate. And that's why it's called the citric acid cycle, because citrate is citric acid. Go ask your chemistry teacher about the difference between ates and acids. And then the coenzyme A is released and can go back to bind to another acetyl group. Okay, so that's just a little circle that happens. Okay, so the citric acid cycle is going to start with this citrate. Notice that we did add water here, so you do need to have water to do this process. It is also going to produce water at several places. So water goes in and out throughout this process. Now, there are so many reactions with so many enzymes involved and the molecules. It's so okay, so we're not going to learn all of this. Okay, you're welcome. What we do want to know is we start off with citrate, 6 carbons, 2 carbons from glycolysis and 4 from the oxaloacetate that is remade after the cycle. And then we are going to add some water. We're going to harvest NADH, and we're going to produce carbon dioxide. Then we're going to harvest some more NADH and produce more carbon dioxide. Remember, we produced carbon dioxide up here in the prep reactions. So we've now, for each pyruvate, we've produced 1, 2, 3 carbon dioxides. How many carbons did we start with? 3. Where did those come from? Glucose. So what happens to all of the glucose that you take into your body? You exhale it as CO2. Wild, right? Like I took in solid food and I exhaled it. Like what? But the energy from the chemical bonds and the energy in the hydrogen atoms is going to get used as energy in our cells. But how cool is that your body just transforms solid things into gases and then you just exhale them? What? Okay. All right. So we make in the citric acid cycle, we make 2 CO2s plus 1 in the prep reaction. We have 1 NADH plus H plus, another NADH plus H plus, and then down here an ATP. I know you're looking at this and you're saying that's a G. That's a GTP, not ATP. Okay, so guanine triphosphate is really similar to adenosine triphosphate, and in different tissues, guanine triphosphate gets made instead of adenosine triphosphate. They can both be used. I know, but for our purposes, it's ATP. Okay, and I know I should have used one that only had ATP. I just wanted to introduce, sometimes GTP is made. For our purposes, it's going to be ATP. Okay, so it's ATP. Just forget you ever heard about GTP. All right, so we make GTP, ATP, and now we're back to a 4-carbon molecule. That molecule is then going to give up another couple of hydrogens. It's going to take some more water. We're going to harvest another NADH plus H plus, and then we're back to the oxaloacetate we started with that combined with the acetyl-CoA and create another citrate, and we do the cycle again. Okay? So those are all the steps. But what you need to know is just this. The citric acid cycle, also called the Krebs cycle. Krebs was the one who discovered it. It's a series of chemical reactions that harvest the energy in the pyruvate and transfer atoms to NADH and FADH2. Oh, that's what this is. This is FADH2. Ooh, I really should have looked more carefully at this. For each acetyl-CoA, we're going to get 3 NADH plus another H plus. We're going to get 1 FADH2, 2 carbon dioxide molecules, and we will have gotten rid of all of the carbon from glucose. And we're going to get 1 little ATP. So for each glucose molecule, we double all of these. 6 NADH, 2 FADH2s, 4 CO2s, and 2 ATPs. That's your Krebs cycle. See? Simple. Okay, so we did glycolysis in the cytoplasm, made pyruvate, took that into the mitochondrion across both membranes, produced some CO2, did the citric acid cycle, made some more CO2, harvested a bunch of hydrogen and attached them to NADH and FADH2. And now we're going to go to the final step and use those hydrogen ions to gather and to create the energy that is going to be used to make ATP, so that we can then use the energy in ATP to do stuff in the cell. All right, so that's the citric acid cycle. Next up, we're going to talk about this electron transport chain.